Today we are talking in great detail about the structure of DNA. And the first thing to note about DNA is that DNA is a polymer. This means that it is made out of monomers, and the monomers of DNA are nucleotides. Now, this is going to be different than our previous learning about monomers because this monomer is actually made up of its own separate parts. So DNA is made up of nucleotides, but nucleotides are made up of three different parts. The first part that we're going to talk about is the phosphate. And phosphates are a phosphorus, bonded to an OH, double bonded to an O, bonded to an O that has a negative charge because it's lost its hydrogen, and then bonded to another O. Technically this is an OH, but that H was removed when this phosphate was bonded to a deoxyribose sugar. So my deoxyribose sugar is made up of a carbon ring with an O in the middle, just like we've done in the past. So I've got a C bonded to a C. Now this is a C that's in the ring. I've got an O at the top. And then there are 4 carbons in the ring because this is a 5-carbon in total sugar. And then this carbon here is bonded to a nitrogenous base. Now normally when we have a structure that we draw and we don't put the C's in, so we draw it like that, normally each corner would be a C, but this is a nitrogenous base, which means that these corners are actually N's. And we're not going to worry about that. That's just a shape that we're going to draw. It doesn't actually represent a carbon ring structure. It's just a shape. So to indicate that this is just a shape, I'm actually going to just kind of shade it in because I don't want you to misinterpret this. Okay, this thing that I just shaded in is a nitrogenous base. And you are going to need to be able to recognize that this is a nitrogenous base. So I'm also going to color it so that there is no doubt in your mind that this thing here is a nitrogenous base. This carbon ring structure that is a sugar has a special name in DNA. It is a deoxyribose sugar. And I'm going to color this deoxyribose sugar red. And then last but not least, we have the phosphate here. And I am going to color the phosphate green. There's no reason why I picked these colors, I just did. So this whole thing is a nucleotide. Nucleotides are made up of 3 parts: a sugar, a phosphate, and a nitrogenous base. DNA is made up of nucleotides, so it is made up of this structure repeated over and over and over again. The backbone of the DNA is called a sugar phosphate backbone. Because it is made up of sugars and phosphates. So here we're going to dive into what actually is a phosphate and more detail about the sugar. So a phosphate, if I draw it in all of its detail, has that OH at the top, bonded to a P, double bonded to an O, and this O has lone electrons. Then the O on this side, it actually has stolen some electrons. So it still needs its outer shell of electrons to be full, but because it stole electrons, it has an overall negative charge. And then there's another OH on the bottom, and that's a phosphate. The sugar is made up of that ring with an oxygen at the top, and then there are 4 carbons in the ring, and then there's 1 carbon sticking out of the ring. Now this carbon that's sticking out of the ring also has an OH. And this oxygen has 2 pairs of electrons. This carbon has an OH. This carbon has an OH. And then there are enough hydrogens to make everything happy. So this carbon is going to be bonded to 2 hydrogens. This carbon is going to be bonded to 1 hydrogen. This carbon has 1 hydrogen, 2 hydrogens, and 1 hydrogen. So that everything is nice and balanced. This is a deoxyribose sugar. When we're talking about sugars in particular, we count the carbons in our sugars. And we do that so that we can talk about where these H's and OH's are located. When we count carbons, we start at the oxygen and we count clockwise. So I'm going to make a little note right here that says counting carbons. And what I'm going to do is I'm going to start at the oxygen, and I'm going to count clockwise. So what does this look like? If this is the oxygen, that is the oxygen that I'm talking about, so it's the oxygen that's inside of the ring, this carbon here is going to be carbon number 1. This carbon here is going to be carbon number 2, because I'm counting clockwise. 3, 4, 5. So this is a 5-carbon structure. When I'm talking about this, we're going to use this a lot, so you're going to need to be able to count the carbons. And it's important to note that on this sugar, there's an OH at 1, at carbon number 3, and at carbon number 5. And the reason that that is important is because anywhere that you have an OH, you can remove it, and you can bond it to an H, and you can form water, and you can do dehydration synthesis. So what ends up happening is when you bond together this phosphate and this sugar, you end up losing this OH, and you lose this H, and these can then bond together. So this bond right here, with this oxygen bonded to a P, bonded to a C, that is this oxygen here. When this comes off, and when this comes off, you get water, and you're able to form a bond between the oxygen and the carbon. We're going to spend a lot of time on that, so we will come back to this later. But for right now, you need to know how to count the carbons. You know that you start at the oxygen that's inside of the ring, and that you count clockwise. So carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5. What some students do is they make a mistake and they start and they count the oxygen as 1. And the oxygen doesn't count because it is oxygen, and we are counting carbons. Moving on to the next page, we're going to talk about the nitrogenous bases. There are 2 types of nitrogenous bases. There's a type of nitrogenous base that is made up of a ring structure where there are nitrogens making up this ring. And then there is another type where you have that same ring structure, but it has another ring of nitrogens sticking off of it. These ones here are called pyrimidines. So if they're made up of 1 ring, they're called a pyrimidine. If they're made up of 2 rings, then they are called purines. And this is a vocab word that you're just going to need to know. The 2 nitrogenous bases that are pyrimidines are thymine, which we call T, and cytosine, which we call C. There are two that are purines. These are going to be the adenines, which is A, and guanine, which is G. The way that I remember which is all gods are pure. And what this tells me is that the A and the G are purines. That's going to help me remember this because the purines are A and G, which means the other ones, T and C, have to be pyrimidines. There's a rule about how these bind together. So the nitrogenous bases are what make up the inside of the two strands, and they're what hold the two strands of DNA together because DNA is double-stranded. And the way that they hold it together is by bonding these nitrogenous bases together. And they bond with hydrogen bonds, and the way that it works is A is going to bind to T with 2 hydrogen bonds. And then C binds to G with 3 hydrogen bonds. So let's see how this works. Notice that if A binds to T, A is a purine, T is a pyrimidine. So we're getting something with 2 rings and something with 1 ring bonded together. And then C binds to G, so again, we're gonna have something with 1 ring and something with 2 rings bonded together. Notice that the T and the C do not bind to each other, and the A and the G do not bind to each other. So you always end up with 2 rings on one side and 1 ring on the other side. It doesn't matter which side is which. It just matters that you're using a combination, 1 ring and 2 ring. So I'm gonna draw my 1 ring on this side, and then I'm going to draw the 2 ring thing on this side. And then below it, I'm going to draw my 2 ring thing on this side and my 1 ring thing on this side. The reason I'm doing this is to show you that it doesn't matter which side has 1 ring and which side has 2 rings. If I put 2 hydrogen bonds, so this dotted line represents my hydrogen bonds. If I put 2 hydrogen bonds here and I put 3 hydrogen bonds here, can you figure out which is which ring shape is which? So those are my hydrogen bonds. And using what we learned, I can tell that this must be a T. The reason that I know that this must be a T is because I know that it has 1 ring that makes it up, which means that it is going to be a pyrimidine, which means that my options are either T or C. I then learned here that A binds to T with 2 hydrogen bonds. So the fact that it is either a T or a C, but it now has 2 hydrogen bonds, tells me that it can't be the C, it must be the T. And if this is the T, then this must be an A, because A binds to T with 2 hydrogen bonds. Now, the other thing that I can tell then is if this one is a T, then this one must be a C, because it's the other pyrimidine, and this time it has 3 hydrogen bonds. If C binds to G, then this one has to be a G. There are a couple of ways that I like to remember these different things. The first weird little rule that I'm going to remember is I'm going to remember the words large and cute. The word large has the letters A and G, and the larger bases are A and G. The word cute has the letters C and T, and little things are cute, so the cute little nitrogenous bases are going to be C and T. So that's one way that I'm going to remember which one is which in terms of size. The other memorization tool that I have is that there are 2 apples in the tree. And why does this help me? Because apples and tree start with A and T have 2 hydrogen bonds. For C and G, I have to remember that in Atherton, a lot of the houses have a 3-car garage. And so for this one, my C and my G bond together with 3 hydrogen bonds. I highly recommend that you go through these notes and you add your own things that you might need to remember or that help you identify these different things. Update your notes with your own information to make these notes better. This is the basics of what you need to have, but you can always add more. Have a nice day.